So hello everyone. So my name is Yan Xia, and I'm from Zip's lab in Carnegie Mellon University. So basically, in the second slide, I just talk about the data collection we have in links, and our goal is to use the links data set to infer the target protein, which we call source, of a small molecules using the level 4 gene expression data. So our hypothesis is that if a small molecule binds to the source and shuts it down, then the downstream gene expression profile should be similar to just using a RNAi to knock down that source protein. So these two expression profiles should be similar. So this is the hypothesis, and basically we want to use the links data set to validate this hypothesis and make predictions in the future. And so the outline of this talk is that I'm going to first talk about how to construct a training data set from links. And basically, so specifically, I'm going to talk about how to, sorry, so the slides are not moving. Okay, yeah. So now, I'm on slide 4 now. Right, so the outline of this talk is first to talk about how I construct a training data set from the links, and so specifically which drug response experiments and the gene knockdown experiments do I choose. And then from this data set, so I'll talk about how I extract features just from these expression values. So generally, these features are specific to one drug, one gene, and one cell line. So throughout this talk, I'll use D to index drug, G to index gene, and C to index cell lines. So basically, this so as you will see later, all of the features. Most of the features are D by G by C. So the dimensions are D by G by C. And then with these features, we want to validate our hypothesis by predicting drug targets using these features. And so specifically, from a drug and a gene pair, we want to use the features that I extracted from the expression values from different cell lines to predict whether the drug targets the protein encoded by the gene. So this is the outline. So first, I'll talk about how to construct the training data set. Right. So basically, we want to have drugs that with known targets, so that we can know whether our prediction is correct or not. So to get a list of the known drugs and their targets, we use the ChemBL data, the ChemBL website, which has the compound ID and their targets. So to cross-reference these compounds with the compound annotations in the links data set, we use so the compound names, the synonyms, and these specific chemical signatures of the small molecule to cross-reference with the small molecules in links. And by this approach, we were able to identify 1,031 out of the 1,300 FDA-approved drugs in links. And for all of these drugs, we know its targets. So we know the gene symbol of their targets. And some of them will have multiple targets. Some of them has only one target. And then, so with these drugs, we want to extract the experiment of applying these drugs on the different cell lines in links. And we use two criterions for the cell lines that we choose. So the cell lines should have lots of drug response experiments and gene knockdown experiments. And we only use the gold experiments because they are reproducible and have higher quality. And for each of the drug, we also need the drug's targets to be present in the knockdown experiments available for the cell line. So So by these two criteria, we chose these 7 cell lines, and so the second column lists experiments they have on this cell line, and the number of knockdown experiments on this cell line, and also the control experiments. And for all of these drugs, their target knockdown experiments are present in this in the knockdown experiments. So from these 7 common cell lines, we chose 29. So there are 29 drugs that are present in all of these 7 cell lines, and 3104 knockdown genes in all of these 7 cell lines. So so in the first phase of the experiment, we only use these drugs that are present in all of the cell lines. But in the second phase of the project, we also have a method to deal with the missing cell lines, so we can work with a lot more drugs. So from these level 4 Z-scores of both the drug experiments and the gene knockdown experiments, so now for each drug, we have drug response experiments from 7 different cell lines, and for each gene symbol, we have their knockdown experiments also in 7 different cell lines. So because there are multiple experiments, so for each of the drug cell line, we just take the median of the expression value. For each of the drugs, we have a 7 vector, which is a 978 by 1. And for each of the gene symbols, we also have a so for each of the gene symbols, we also have a 907 by 1 vector, one per cell line. So basically, this is the data we extracted from the links dataset. And now using this level 4 Z-scores, we want to construct a set of different features to capture the idea of the correlation between drug experiments and knockdown experiments. And I list them here, and I will explain them one by one. And again, they are specific for each drug, for one drug, one gene symbol, and one cell line. So the dimension of most of them are D by G by C. So just to remind, the basic hypothesis is that so the small molecule turns off a protein, then the downstream gene expression profile should be similar to just doing a gene knockdown of that protein itself. So we have the data basically for each of the drug. We have 7 vectors of length 1,000 by 1 basically, and for each of the gene symbols, we also have their expression vectors in 7 different cell lines. So basically, for each drug and each cell line, we just compute the correlation between the drug in that cell line and each of the 3104 gene symbols, gene knockdowns in that cell line. So for each cell line, as you can see so for each cell line, we have a 3104 by 1 vector, and because there are 7 cell lines, so for each drug, we end up with a 3104 by 7 vector matrix of correlation values. And because we have 29 drugs, so that's our D by G by C matrix. So this is the correlation feature, and we want to see if this correlation number has some predictive power. So basically, so for each of the 3104 gene symbols in different cell lines, we rank them according to their correlation values. And for each gene symbol, we choose their best ranking across 7 different cell lines, and use the best ranking for that gene symbol. And using this ranking and using this best correlation number to rank the gene symbols, we want to see if the correct target of the drug is present in the top 100 list or not, in the top 100 genes or not. And as summarized in this table right here, for the 29 drugs, we're able to pick 11 out of the 29 drugs that has their targets in the top 100 list. And so here's the mean of their rankings. This graph on the right basically shows the comparison of these experiments with the random experiments. Each dot on the red curve represents a drug. Yeah, and basically, so for each dot, it represents the rank of that drug, and how many of the drugs can be selected if we use the rank of that drug as the cutoff to say this is the positive target. So that curve is generated using the target genes, and the black dots are generated using just by randomly choosing genes with the same number of the targets for that drug. And we see that for some of the drugs, this effect, so this correlation feature seems to be effective, seems to be statistically significant. But because we're doing this across different cell lines and 7 different cell lines and the different number of targets, then because of this is like a multiple testing procedure, so we can see for some of the drugs, this correlation procedure does not seem to be very significant. But overall, we think this feature, this is a useful feature of picking out correct targets. Okay, so this is the first target's correlation, and the second target we called it cell selection. Yeah, so correlation was the first feature, and now we're on to the second feature, which is called cell selection. So basically, we have features from 7 different cell lines, but some cell lines are maybe more helpful than the other cell lines, and we want to somehow weight their contribution higher than the other cell lines. So if you just calculate the correlation between a drug response and the cell lines control experiments, we expect to see that the favorable cell line have a low correlations with the drug experiments. So basically, we want so after the drug treatment, we want its gene profile to be much different than the control experiment. And we think if they are different, these cell lines more useful. And that's the basic hypothesis. So we have the drug response numbers for each for each of the drugs and for different cell lines. And for each of the cell line, we also have its control, the median value of its control experiments. So we basically calculate the correlation between the drug experiments and the control experiments. And for each drug, we have a 7x1 vector. And this feature is not a gene specific. So for each drug, we just have 7 values for different cell line. Right. So the third one is a protein-protein interaction correlation score. So the idea is very easy. So because our hypothesis is that the correct the source should have high correlation with the knockdown experiments. So by the same logic, we think that the direct binding partners of the source protein should also show high correlation. Should also show high correlation with the target's knockdown experiments. So basically, we call this the PPI correlation score. And to calculate that, just focus on this orange box right here. This represents one gene symbol. So this represents one drug, one cell line, and one gene symbol right here. So for this gene symbol, we look at the PPI graph and find all of its neighbors. And basically, we want to calculate how many of these neighbors are in the top 100 list sorted by the correlation score over the intersection of their neighbors and all of the genes. So we think if this number, so the higher this number is, the more relevant this gene symbol is. And from this procedure, we also generate a 3104 by 7 by 29, which is the number of drugs features. So this is the PPI correlation score. And we look at, just by using this PPI correlation score alone, how well can we predict the correct targets, which is showing here. So basically, we just use the same procedure. For each gene symbols, we have for each gene symbols we have 7 different PPI correlation scores, and we sort the gene symbols using these PPI correlation scores. Basically, we choose the best PPI correlation scores across different cell lines, and we sort the genes using the best correlation scores. And we want to see whether the correct targets, the correct targets for the drug is in the top 100 list. And just by using this PPI correlation score alone, we're able to pick out 10 out of the 29 targets. And the mean ranking drops. The mean ranking decreases from using the correlation features. Basically meaning that building on this PPI correlation feature on top of the correlation score, the raw correlation score can help can enhance the predictive power. And then we also use this PPI protein-protein interaction graph to construct a different type of score. And this is the idea from Avi, and it's very similar to the correlation score, but we're not looking at the correlation matrix anymore. So we're directly looking at the drug response experiment, and the hypothesis is that the neighbor of the target of the drug should have a high expression value because their binding partner is being suppressed. So they should have high change of the expression value from 0. Because we're dealing with the level 4 Z-score data, so we just want it to be different than the mean number. So to carry out this idea, again, for each of the gene symbol, we find its neighbors, and we look at their neighbors present in the landmark genes, and we calculate either the mean of their PPI, basically calculate the mean of these neighbors' expression values, and the max of the absolute value of their expression values, and we have two different matrix corresponding to this feature right here.
I don't think you should use the landmark. I think for that one you should use the entire expression, the extrapolated.
Oh, okay. Yeah, because the others are computed based on the landmark. So after we build all of these features, we want to combine all of these features and basically train a classifier to predict the target from these features. And now, what we have right now is that for each of the drug, we have all 7 cell lines, and for each of the cell line, we have a set of 4 features. But because the last feature has 2 vectors, so so for each of the cell line is a 3104 by 5 matrix, and we have 7 of these. This is basically the feature matrix we have collected so far, and to construct a training set for the classifier. For each of the drug, we pick out the features from all of the features from their targets, which is colored in orange here, and their corresponding label is 1, means it is a target. And for the rest of the genes, we random sample 100 negative genes from the 3104 minus the number of targets genes, and we append their features, and their label is 0. So the training data set for one drug is the number of targets plus 100 times 7 times 35, because again we have 7 cell lines and the 5 features for each cell line. So this is the training data set. And to use this training data set, we first tried to train a logistic regressor using this training data set. So we use the leave one now cross validation. So basically, we train a logistic regressor using the 28 drugs, and test all of the for the held out drug, use that trained logistic regressor to test all of the genes, and sort the genes and rank the genes by their predicted probability from the logistic regressor. And then, so here's the results showing in the third column. So using the logistic regressor, so the logistic regression is able to pick out 11 out of the 29 targets in the top 100, and the mean value of the rankings dropped as a little bit. And then we think that so the decision boundary of a logistic regressor is linear, and it assumes that all of the features are independent. So next we tried a random forest, which is very easy to apply, and has a more sophisticated decision boundary, and it also offers data result interpretability and automatic feature selection. So yeah, so basically we just built 2,000 different decision trees, and we use the same leave one out cross-validation procedure to test this random forest, and the result is much better than the logistic regression, in that we're able to pick 16 out of the 29 targets, 16 out of the 29 drugs, and the mean ranking dropped significantly from logistic regression, and from using the features individually. So basically, what we learned now is that random forest seems to be a very good technique, and we have the results for the 29 drugs and the 3104 genes. But these 29, so we were limited by the 29 drugs because they're the only one that has experiments in all 7 different cell lines. So what I said was we were limited with the 29 drugs before because they are the only one that has experiments in all 7 cell lines. But there are a lot of more drugs that we can test, but they don't have all of the data. So actually, there are 152 drugs have a subset of at least 4 cells from that 7 cell lines, and so basically they have different combinations of that 7 cell lines, and we need a way to deal with. So in order to apply the random forest classifier to more drugs, we need a way to deal with these missing data, and unfortunately, random forest automatically provides a way missing data because it does not require all of the features in one decision tree. So we came up with the 2 ways to deal with this missing data issue, and the first is very naive, and the second we think is better. So the first one is that so basically construct a training data set on the fly. So say we're testing a drug 1, and the drug 1 has BCD 4 cells, and we basically just look through all of the rest of the rest of the drugs and see which drug has the cell line profile that's compatible with the drug 1. So say drug 2. So for drug 2, we just pick out the corresponding columns that correspond to BCD, and for 3, we're missing AE, so we can't use that. For 4, we pick out BCD again. So for each drug, so when we do leave one out cross-validation, we can construct a data set on the fly, and train a random forest and test on that held out drug. And just to carry out this, just to test this idea, we're able to pick out 58 drugs that has the target ranked in the top 100, and which is 38%. And we see that so the more cells, basically the more cells we have, the more information we have, the better ranking it is. So the higher percentage we can pick out. And so for 4 cells, we can only pick out 27% of the drugs with 4 cells. And just to show that this procedure, so compare this procedure to a random experiment again. Right. So basically, instead of using the correct targets, we basically use the random genes as their targets, and we want to see if we use random genes, how many drugs we can. So what's the fraction of drugs we can pick out? Basically, the mode is at about 0.075. So basically, it's 7.5%, and our result, 38%, it's much higher than the random experiments. So basically, we're thinking our random forest classifier is picking up some meaningful signals in our data set. So this is the first naive strategy. Yeah. So the second strategy is basically just to construct a unified random forest, and the idea is that we want to construct a random forest. So it's like a hierarchical random forest. So we want to construct a forest with 3,500 trees. At the first level, we just pick 4 cells. We just pick 4 cells randomly, and for that 4 cells combination, we look over all of the data, all of the drugs, and then find ones that's compatible with these 4 cells. And we train the tree. We train one decision tree using all of this data, and we repeat this procedure for 3,500 times basically. And for each tree is a independent random selection from the 7 cell lines. And we use 3,500 because by chance each combination will have 100. Each 4 cell combination will have at least 100 decision trees. So this is the second strategy, and we did the same analysis using the second strategy, and we compared that to the first naive strategy. The second one turns out to work better than the first one, in that it's able to pick out 64 out of 152 drugs, and that's 42%. And if we only look at 5 or more cell lines, it's basically 50%. So we're able to pick up 50% of the drugs if we only look at 5 cell lines or more. And also, if we look at if we be more stringent, we don't look at top 100, but we categorize top 50. So if the target's in the top 50, we classify them as a target. Then this unified random forest is able to pick out 54 drugs, which is 34%, which is almost as good as the 100. Basically, it's almost as good as the top 100 result from the on-the-fly data, which is the 38%. So basically, we think this second strategy is much better. So basically, what I showed is that by selecting good experiments from the links data set and construct different features using the raw expression values, level 4 expression values, we're able to test our hypothesis. It shows that our hypothesis is able to pick out targets. So it's able to pick out targets for a drug. So in the future, our idea is to apply this random forest classifier to a unknown drug. So so there's a drug we want to know its targets. Basically, we just need to collect its gene expression values in different cell lines and construct the same feature as what we did now, and we're able to predict which gene symbols might be its direct targets. And so that's what I've been doing right now. It's basically applying this random forest classifier to small molecules without known targets. And we also want to further narrow down the top 50, the top 100 or top 50 list to probably like top 10. So we're working with Dr. Camacho from University of Pittsburgh, doing some structural analysis. Basically, it's just doing small molecule protein docking analysis to basically further rank our hits by their interaction scores from their docking models. Yeah. So that's all. Thank you guys very much.
Thank you, Yan. Please unmute yourself if you would like to ask a question, because I may have muted you inadvertently.
Okay.
I have one suggestion, and there is this data from Peter Sorger's group. It's also links data. It's called the kinome scan and native scan. And in there, they did some experiments for kinase inhibitors, and they have affinity scores for kinase inhibitors with their expected known targets and also off targets. So that would be also a good data set to validate your predictions.
Okay. Yes.
Do you know how many cells they have and how many?
I think they did a lot of the same cell lines and the same conditions as links. So There is also, within links, there is a dataset called LGP. That is a links joint project. And in those links datasets, they use the same drugs.
And what about, so obviously the real thing we're interested in is the small molecules. So the drugs aren't drugs, they are not.
Yeah, the small molecule.
Small molecules.
So there is about 200 kinase inhibitors that are considered small molecules, and there are some of them.
Intersect the links one?
Yes, it's part of links. You could talk to Chownan, who processed that data from our group, and he can give you the access to both datasets. So
Okay.
if you send emails afterwards.
Yeah, this is Mustafa. I worked with Kayi from the University of Washington, Tacoma before.
Okay.
Yeah, I have a question for you.
Okay.
Thank you for the presentation. It was quite interesting. I was wondering whether you take into account the different dosages and measurement time for the drugs, and for the knockdowns, they also have measurement times, right?
Right. Yeah.
Yeah. Did you consider that, because the profiles can be quite different between dosages?
Yeah. So, yeah, I agree. Like currently, our way to basically construct that basically it's like a signature for each drug is very naive, I would say. Basically, just take the median of all of the experiments. I'm just thinking about how can I incorporate this dosage information, maybe just incorporate this dosage information somehow into our features and time points. How do I deal with the drugs with multiple targets? Yeah, right. So currently it's a little optimistic. So basically we just use the so if the drug has multiple targets, we just use the best ranking of one of their targets as a result.
I see. So one with the highest ranking between.
All the randomization tests take that into account as well.
Right.
So all the analysis we do with random data assumes the same number of same distribution of targets as the real data.
Yes.
So we are not trying to get all of them, but we are trying to get at least
one of them. Yes.
I see.
Because
right.
Yeah.
In the result that you showed, you probably put the best or the highest ranking between these targets, right?
Yes.
Okay. I see.
Actually, I think most of them only have one, no?
Yeah. So the majority of them has only one target. Yes.
I see. Right. Thank you.
Yeah. Thank you.
Hi, this is Stefan. Can I ask a question? One comment first. So all drugs have many different targets, right? So if you use FDA-approved drugs, then you can assume safely that they all hit multiple things.
Yeah.
But I have one other question.
Well, but at least the database lists one.
Right. Yeah.
Well,
I agree, but
that may be the case, but nonetheless, they hit multiple targets.
yes.
So maybe the results are pessimistic. Maybe we actually get much better results, and we just don't know that these are the real targets.
Right.
That you're getting something. So I was wondering, did you look at, and maybe you should try to look at, evaluate the predictive results based on the type of drug target, right? Because you would expect, right, that knockdown RNAi would work much better. Would be much better correlation to a drug for some targets versus others, right? For simple enzymes, for example, you could expect that maybe there is some correlation, but there could be others. For nuclear receptors, I mean, you wouldn't really expect any kind of correlation, right, between knockdown.
So you're saying basically divide the 1 and 2 drugs in different families depending on the type of targets they have and see how we perform on each of them.
Yes, because you would not expect a correlation, right, for some drug targets. It's not a very good correlation.
Yeah.
Very nice.
This is Roger Baumgartner. This may sound like a silly, simple question, but for the RNAi experiments, did you filter those at all by the ones that were shown to actually knock down the gene that the RNAi allegedly targets?
Sorry, could you repeat your question again?
So the RNAi is allegedly targeting a certain gene, but a reasonable subset of those experiments, the RNAi doesn't do a very good job of knocking down that gene.
Okay. Okay.
So you could, in principle, filter out the ones where you don't think the RNAi has done what it was supposed to do.
So you have a list? You have a list for those?
You have to look to see if the RNAi actually knocked down the gene it was supposed to knock down, or if it's targeting something that's not in the links that you have.
No, I understand. Did anybody look?
Yeah, I don't have a list of those myself, but it's relatively easy to produce such a list by looking through the data.
So are you going to produce it?
Well, I know that that's one of the things that Hong and Kai's group is looking at. I think he's on the line.
Hong, do you have a list?
Hi, this is Kai Young from University of Washington. We have been looking at level 3 data. So we are not looking at the same set of cell lines, the 7 cell lines that you guys are looking at. We do have a list for our own, the subset of cell lines we are looking at, but I am not sure, I don't think it's the same list of cell lines that you're looking at.
But is there a method to give you a cell line that you can generate, or is it very specific to this?
We just look at if the genes got knocked down itself. So if you are trying to knock down gene G and see if gene G's expression level is close to 0.
Right.
Yeah. Okay.
Oh, okay. Okay, so that's
Yeah. Yeah.